About what has been transformational for me about systems biology, and I started thinking about it in the middle 80s, is turning that same kind of logic to medicine and to disease, and realizing that really what disease is a biological circuit, one or more, that have been perturbed by mutations or been perturbed by environmental signals, and that alters the information expressed, and that leads to the nature of the disease, but it leads to more. It leads to fundamental new insights into how you do diagnostics and how you do therapy that are really, as you'll see in a moment, quite transformational. Here is a vignette, a snapshot of 3 different time points in an important biological circuit in the brain in mice that have been infected with disease-causing prions. And the little red dots mean that there are proteins in the diseased mice that have increased their levels in really striking fashion. And so you can see the panel on your left, there isn't much change there. In the middle, there are a few changes. In the far right, that's just a few weeks before the animals are actually die of the disease. So there are really 2 interesting questions we can ask. If we look at these networks, we can ask ourselves, won't the future of drugs be picking targets in these networks which, if perturbed by the drug, will make the network behave in a more normal fashion? So the logic of drugs isn't going to be a list of 5 major types of drugs that everybody uses now and pick one of them and see what you get. It is the logic of modulating disease-perturbed biological networks. And the second point that's really interesting is you see very early in that middle panel, proteins have changed far before any clinical signs come up. And so the interesting question you can ask is, if that protein is actually secreted into the blood by the brain, can we use its quantitative change to predict the disease? And it turns out that the answer to that really is true. And we are in the very earliest phases of proving the things I'm going to say next, but I'll guarantee you they're going to turn out to be true. There are of the order of 50 tissues and organs in the human body. Each of those organs secretes proteins into the blood that constitute a molecular fingerprint. The relative ratio of those proteins reports the state of the circuitry that's present in that organ. Hence, there'll be one relative set of values for a normal liver, and there'll be a different set of relative values for a liver infected with hepatitis, or a liver that has cancer, or a liver that's cirrhotic from drinking. So the idea then is, if every organ puts these out, we can read these 50 molecular fingerprints, and we can assess health and disease on a regular basis. So what is the real challenge in doing this? And I won't tell you how we discover all these things. The real challenge is, how can we measure 2,000 or 3,000 proteins from a droplet of blood? And what we have done in this regard is set up a major collaboration with Jim Heath at Caltech to use nanotech chips to do these measurements. And the idea is that we will indeed, within 5 to 8 years, be able to take a fraction of a droplet of blood and make those 2,000 measurements, and I'll tell you why that's going to be important in just a few moments. What Jim is really pioneering are nanowires. These are small little wires, each of which can be individually functionalized to capture and report on a different protein. He can make 2,000 of these nanowires in a parallel array that spans less than the diameter of a typical eukaryotic cell, 10 microns or so. And it is these kind of chips that we're going to use to read out the future. They will be printable, manufacturable. You can use them and we'll produce them at very large levels, and the measurements will become enormously inexpensive. Now, the other technology that's going to be key for this new kind of predictive medicine is sequencing your genome. So we're now working with a company in Boston that is using nanotechnology principles to determine the order of letters in single strands of DNA. And what is really striking about the potential of this company is that it will have the potential for sequencing simultaneously 1 billion strands from your own genome, doing it very rapidly and very inexpensively. And of course, that is going to be the basis for inexpensive genomes that will be one of the initial foundational points in this whole idea of predictive medicine. We'll talk about that in just a moment.